Okay, yeah. So basically, I've been in love with this group of frogs. I mean, like I was not studying this when I was undergrad, but then I start studying this group of frog when I did my PhD, basically. So this is the Asian ranid frogs with Gastromysophorus tadpoles. Yeah. So then like Huia, what I'm going to talk about, like the focus of this talk is actually just one of the three of Gastromysophorus tadpoles genera at the time, prior to my studies. Yeah. And then, so what is Gastromysophorus tadpoles? It's a little tricky to pronounce, yes, that's also Harry. But then this is basically a belly sucker. So that's easy to remember because look, if you can see, this is the sucker that they have. So this is like a specialty of this group of frog because there's no other frogs that like develop this kind of adaptation among other anurans. And so this is just in this group of frogs who are in Asia, and another one is like a neotropical buffonid. So what's so special about this? So they develop this abdominal sucker because they are living in a cascading habitat, so fast flowing water. So to adapt, to survive, to make it to an adult, they develop such a unique adaptation. So how is this sucker work in the natural habitat? Here I can show you the video that I took in the field while I'm doing field work. So, and then it's been featured in the Discovery Channel Canada. So as you can see here, this is the tadpoles. So they attach here using like this sucker that they have in their belly. So basically to against this, and then you see how strong this current is because this is like really literally like below the swift. So this is really strong. But guess how, like, do you think like this frog moving, do you think they use the sucker? No. No. Look at this other. Oh wow, this is not working. The other video is not working. So basically this this sucker here, it's actually literally just to hang on the rock, and to move they use their mouth part. This is how amazing this tadpoles are. So they're basically like eating while dragging their, like the whole body. Yeah, now you can see it. Yeah, see, that's how incredible this tadpole is. So this sucker is really strong, and then it's literally just to hang in, as you can see in the previous video. And then to move, they're basically using their mouth part, like grabbing like the algae surrounding the surface of the rocks, and then just like dragging the whole thing because they still need to survive, not to wipe out by this water. So now let's have a look. How is the geographical distribution and species of this frog? So Huia was described in Borneo, and then they in 1893, and then the next species that they describe is the one from Java, Huia masonii, and actually it was like Javana, but then through study they always like have some contradictory, whether like Javana or and masonii are like two separate species, or not. But then like prior to my study, they're like synonymized. And the next species they describe later on is in 1991, which is in Sumatra. And then they also describe Huia modigliani, but this Huia modigliani never, never been recorded after the original description. And there's only one species in the mainland, which they call it like Huia melasma, which is in 2005. Now, see, considering like this large range, like wide range of geographical distribution of this group of frog, and for me, it's like just doesn't make any sense to have only like 5 species within this genus, especially when we have to compare it to like the other 2 genera that I mentioned earlier. So Amolops is in the mainland of Asia, and then at the time it was like 55 species, and Meristogenys especially, it's like 13 species. This is the Bornean genus endemic, and then this 30 species, even they only describe it from the Malaysian part, not even like go to the Indonesian parts. So then This, we leave that out, but then we will just like focus on this group. So the species is really poor. And if you're looking at the phylogeny before my prior to my study, here I just like show you like several studies that included like the species in their phylogeny, and they are very here. But then if you can see, so this is basically all the Gastomyzophorus tadpole, basically like the Huia. But then as you can see here, Huia is actually like paraphyletic in all studies, because look, Huia cavitympanum, the type species, is actually closely related to Meristogenys, the Bornean endemic, other than the other Huia. And then also like there is Clinotarsus here, which is the mainland Asian genus that possesses no Gastromyzophorus tadpoles. So that is another question that I wouldn't really like touch down in my talk now, because we want to focus on Huia. And so what is it? So my hypothesis was, oh, this is probably too few samples. And then when I read carefully, yes, they only included like one individual per species. So that is just like not really representative, considering the large geographical distribution that I mentioned earlier. So in this case, I was thinking whether the Huia paraphyly would be solved if we add more samples. And to do so, as an Indonesian, and then I'm limited with my PhD time. So I was just like focusing on Sumatra. This is the island of Sumatra. And then I mentioned earlier, this is like the mainland part of the mainland. This is Sumatra, Java, and Borneo. So my study was just like focused on Sumatra. Why I choose Sumatra? Because Sumatra is topographically amazing. Because you see this is basically mountainous region. As you can see also here, like this is like from the north to the south. So with this, you can tell that there are so many habitats that are like suited for this kind of frog. But as far as you know, that in that habitats, you only find this like a Odorrana, like the genus Odorrana, or Huia, or Chalcorana. At the time, it was under Chalcorana. And then it was also like a debate whether they're like a Chalcorana crassiophis and companies are two separate species or like synonym. And at the time, we already know that Odorrana is not possessing possessing like a tadpole with a belly sucker, and only Huia that was confirmed to have Gastromyzophorus tadpole. So then there's a candidate kind of, like we're not sure whether like this person, this frog, possessing Gastromyzophorus or not, because there's like so much problem in the taxonomy for this frogs. As you can see here, this is at least so they're actually like abundant. So then whenever I go to this kind of habitat, like they're always there. But then actually like at the time, the taxonomic position was unstable. It has been moving around like here and there, So before they put it in Chalcorana, they put it one time in Phylogenetic and some other genus. But then it's actually like those study who placed them here and there, they never really included the actual specimen of this frog. So then it just as an implication of the other sister group, basically. And then so it's not really know where is it exactly in the taxonomy. But then the other problem is that the type specimen of this Chalcorana company that probably synonymized or like a valid was lost. And then, yeah, no molecular data as well. And then, so there's like a lot of problem. So then in that case, I kind of reform, okay, not only I wanted to see whether like adding Huia samples would change the Huia paraphyly, but then I would also like to answer like where is actually, like this Chalcorana crassiophis of company was in the phylogeny. So I did integrative taxonomy approaches with ecological distribution and, like the frogs and as well like a genetic, but then mainly I try to have the large geographical samplings through fieldwork and through lone specimen, but not only from adults, but also from larvae. Because so far, people only studying the adults, but then like really leave the larvae behind. So it's kind of like a not okay, because especially in this case, it might be, you know, like one of the main character for like defining the phylogeny of this adult. So this is the site that I went to have a fresh collection to add like this samples. And so I collected here like for almost like 1 year. I went to the field in total, and then I went like basically to all possible habitats that I could go, from the north to the south, plus a little bit of Java. And then I collect like every single frog, kind of like that, which is like chalcorana or like Huia that wasn't, but also like the tadpoles. And I did some molecular and morphology to construct the phylogeny. And then I also, like, as I said, like doing some qualitative and descriptive phylogeny. And so this was the previous phylogeny prior to my study. And so what was my study contribute to this phylogeny? So it was just 2 species within Sumatra. So one in Java and then one in Sumatra. But then my study actually like showed that there are like much more diversity within this group. Even I only like added, you know, like focusing on like Sumatra and Java. So through my study here, it's actually like at least in Sumatra, we have like at least 3 species, 3 lineages, like distinct lineages in Sumatra, and then like 2 distinct lineages in Java. And then, interestingly, so where does Chalcorana crassiophis sit? So this Chalcorana crassiophis is actually nested within the gastromysophorus clade here. And then this is like a completely far away from the Chalcorana. So, and then they also like have more than like one distinct lineage here. So, and then that's why we decided to, you know, like erect this as a new a genus. And then the Wijayarana also, like I will explain you in my next slide how we come up with like, you know, naming this as a new genus. So here we go. If you expanded the phylogeny of just like the crassiophis group, this is like the whole individuals that I collected. So they're like here. So the first evidence that it was not Chalcorana was because we collected the gastromysophorus tadpoles that is actually like matched with the adult here. And then as you can see, we also have the topotypic specimen here. And then among tadpoles, you know, that is already like 3 different things, like which is like matched with the phylogeny that I had, but it's also supported by other morphological evidence. This is just like some of the evidence, like from the color, eye coloration, and then this is the nuptial pad, like this first 2, and then like this one is actually like split, but then also like from the morphometric. So with this, we also have an evidence from their geographical distribution. So this basically all of this evidence kind of like supporting that this is like a really independent from Chalcorana crassiophis, and this is, you know, like have enough evidence to delimit this as a new genus that we call Sumatrana. And so what about the Huia itself? The Huia, so this is like if you expanded the phylogeny of Huia, then this is what we got, that like this is actually not really changed the phylogeny, because through my study, the Huia cavitympanum here, the Bornean one, is actually still closely related to Meristogenys other than the rest of Huia. But then as I mentioned before, like the Huia in Sumatra and Java are actually more than what we thought before. So from my result, I had some topotypic specimen, which is like confirmed that Huia sumatrana is actually like in this clade. And also the most important thing is that we resurrected Huia javana. So as I said, Huia javana and Huia masonii was like kind of like synonymized, but then through my study, I had like the sample from from the type locality of Huia javana, and then like with the morphological evidence, it also showed that this is like a valid species. But then we have also like this other lineage here, which is in Java. But then this is also actually like not masonii through the morphological evidence. So our p distance kind of like support like the topology of the tree. And then what's next? So like morphologically also like supported. So this is like especially like from the tadpoles, as you can see, you see the difference between the population in Sumatra, Java, and the mainland. Especially at the time. At the time, we still do not know whether it's has customized for tadpole or not. But then this is within the clade, and it's like here, nested here. So through this evidence, kind of, so and then to stabilize the phylogeny that was unstable before my study, so we decided to, like, according to this evidence, then we decided to just retain the genus Huia for the Bornean population, because that was the type specimen, kind of. And then so to accommodate the other population, we erected a new genus called Wijayarana. So now what my study contribute to this overall knowledge about the Huia and the Gate sister group. So in 2023, yeah. So basically, like, no, Amolops is expanded. Like other people also like study in the region, and they expanded the Amolops into like 80 species now. But my study definitely like adding, like changing the phylogeny and the diversity of like this group. Okay, so Wijayarana is still like 5 species. For now, with some unidentified lineages that I can explain a little bit later. And Meristogenys is still 13. But I also like added like sumatrana that was not really known that it has gastromesophoros tadpole. So this is just like the summary how the genera was expanded from 3 into 5 through my studies. So let's go back to Huia and Wijayarana now, because it's like a new kind of like valid species. So Huia cavitympanum is like a monotypic genus kind of here for the moment. We do not know whether they have more diversity or not. Then I can explain you a little bit later through my future further study after that. But then Huia javana is now valid kind of, and for the moment, we still include like Huia masonii, although my lineage showed that like that unidentified lineage might not be a masonii. But sumatrana is also like confirmed, but modigliani also like still in question, because we still do not know, like have no more evidence whether like Huia modigliani is in one of those identified lineage that I have. And for Huia melasma, the biggest problem is also like the. So then with that, I kind of like, have a further question, which is like kind of like, okay, whether Wijayarana melasma is like has gastromesophoros tadpoles or not, and how species they are, because we never know, and then I don't have access to that. And for Huia cavitympanum, is it really a monotypic genus? And then especially we need to identify this unidentified lineage within the Wijayarana itself and answer the question whether Wijayarana modigliani is a valid species or not. The same goes with Huia masonii. And then, so this research I try to answer through Marie Curie postdoc that I'm doing now. And I basically using the same approach, integrative taxonomy, but then I didn't do any fieldwork anymore. So then, because the main important part was like to have the sequence from the type materials to really like see which one which, to be able to identify the unidentified lineages. So I did also like some more morphological examination from adult and tadpoles. And so what is the progress? So my Marie Curie was started when the COVID hit, then it's been like delayed. And then so that's why I'm currently just got my my sequence. And then so like I couldn't like show you like the final version, So this is like some analysis that I could like get up to know. But from this, basically, as you can see, the general topology does not change. So either using the Sanger sequencing technique or genomics, it doesn't really change the phylogeny. So it's basically, this is what it is. And then, and then, yes. And then, so what about the tadpoles evidence? So I measure much more tadpoles, but then like one of the main finding kind of that also like really like make me happy, because I could really like, you know, like answer one of my biggest research question, especially for the tadpoles from the mainland. So this tadpole that I show you here is actually a Wijayarana melasma tadpole. So I sequenced the tadpoles for this, and then it's matched with the adults of Wijayarana melasma. And that's why it's confirmed that, you know, like this is actually like a Wijayarana, like part of Wijayarana, like the same as the one in like Java and Sumatra, because it has this M beak shape in the upper part. So through my study, I could kind of like easily distinguish that like, yes, tadpoles is the most important character to identify this genera. Actually, like the genera, as you can see here, this is, you can really easily has the pattern, because like Amalops, the mainland population, the beak shape, actually the upper and then the lower beak, if you can see like from these different genera, they are like really different. So the Wijayarana melasma, because it has like M shape here, and then it's like exactly the same as this one. So we can confirm that this is like actually the genus of Wijayarana. And then, so I also have a set of data from tadpoles from Borneo. I haven't really like, you know, have the chance to examine the adults, like more adults from this region. But then from the tadpole collection, as you can see here, I found out that there are at least like 3 morphotypes. I mean, if you see closely like the shape, like the morphology of like this tree, they are like look different, especially in here. You can see that like the gastric, the belly sucker are like different within this tree population. So here kind of like lead me to my first hypothesis that like Huia cavitympanum might not be a monotypic genus. It's probably like has more species within that. I just need more samples, and at that moment I don't have that. So what's next then? Maybe in the future I can come back and tell you, but then that's all for now. You know, like that the application of integrative taxonomy from adult and larvae, like with large samplings, will really like help you to solve the problem, like the phylogenetic problem of like certain species. And so with this, I would like to acknowledge everyone, not only like a funding sources, but then like everyone, because without them, my research that I present here wouldn't be possible. But other than that, I would like to acknowledge myself. So like knowing like who I am and where I am now, because I was born and raised in Indonesia. So I spent like half of my life in Indonesia. I studied in this university in Java. And then from that, because I'm studying frogs, thanks to frogs, I could move to Germany and doing my PhD there, and then doing my first postdoc here there. And also like with that, I also like got the Marie Curie to do a postdoc like in Germany and also the US. And it has been an incredible journey, but it's also not an easy journey. There's up and down, but then I'm really happy because I really have like meet so many incredible peers and women, you know, and then including like this 2 other colleague friends of mine. I knew them already like more than 10 years now. And then so we race and we have like different upbringing because we came from different countries. So I'm from Indonesia, it was from Mexico, and Ceylon is from Taiwan. And then so we all like early career scientists, and then we still trying to, stay in our path, and then we overlapping some struggle everywhere. So regardless where we are, and then that's why we kind of like, okay, join forces together to, let's say, having this initiative to connect women herpetologists across the countries and cultures through the project that we call Global Women in Herpetology Project. And yeah, so basically the goal for this project was to support women herpetologists in all career stages across disciplines, regions, and cultural background. And especially we also wanted to, learning from our own experience, we wanted to encourage the next generation of herpetologists to just follow their dream and without worries. And in this case, of course, we wanted to highlight this because we want to increase the diversity and visibility of women in the field herpetology, specifically, as Angelica mentioned at the beginning, that it's still like male dominated. And we have 2 programs in this, like a book that will that I showed you here. Maybe you can look at this now if you want. And yeah, so the book is a story compilation. This is what I'm going to learn. But then the network directory is basically an online database. So if you identify yourself as an herpetologist and you're like a woman, you can simply go to our website and register yourself. And this is just like, we provide this just for a networking, let's say, student looking for prospective supervisor or collaborators, or and this is also like not specifically for women. Yes, the lists are only like women, but then it's also can be used by everyone, regardless who you are. Okay, so let's have a look a little bit more in the book that I passed you around. So what is this book about? So this book, Women in Herpetology, we call it this, and then we give this title 50 Stories Around the World because it's written by 50 females from 50 countries and regions all over the world, this is already good because it's representing a quarter of the total countries and region recognized at the moment. And so what is the aim? The aim is like to put a spotlight in the diversity of women working with amphibians and reptiles in various profession and career stages, basically. So we have early career authors and we have like late stages career authors, and we have different cultural background, and basically it's an incredible stories. You can see it yourself, and then like you can read it yourself. But it's also like amazing that through this project we could really like collaborate to an incredible artist who also come from different continents, and then like made all of this like a beautiful illustrator that you can also find in the book. And we work for free, so this is a free project, yes, but we really wanted to have this. It's not for sharing. Yes, this is sharing is good, but then as I mentioned earlier, this is actually like for scholarship because we want to make a real action. Like sharing is good, but then if you want to change something and then like do the action, and then so our action was like to collect money from this book to fund like a student to attend herpetological conferences. So this is just like the page that you could find. And then if you see, there's like, this is so amazing. I mean, like we didn't even think about it. That actually like the 3 authors we have are from CBO. So we have Ninda here. And then we also have Raquel and Angelica here. So I, on behalf of like the whole team, really thank you for the 3 of you to, like be willing to contribute to the project. And it's really meant a lot. And yeah, so this is just how the story look like. So basically the story is about like whichever story, like each female's authors we have in the book want to share to the public. We don't restrict it anything. You should tell like this part of the story, so then like it just like we made them free to choose whichever sides they wanted to share to the public during their journey to like pursue their career in the field of herpetology. So this is one of the author from China, and then like she decided to share like her story about like a life in herpetology. So she showed like how she first encountered, you know, like in the field herpetology, and then like how the field work was, how her peer treated her in the field, and many other more. And then the illustration here is made by Huia. She's from Malaysia, but then like has a Chinese descendants. And here's the story from the 3 wonderful females at CBO. You can read it in the book, and then you can also like ask them further after you read it, maybe if you're curious. But then we actually also like ask, you know, like, because, okay, yes, we're happy they're contributing, but we wanted to know why they actually like wanted to contribute. So here we show you just like the 2 of many people responded. So this is like our author from Ghana. And then she said, I wanted my story to be heard, to inspire young women, especially those in Africa. And she highlight that like we can make it if we put our minds to it. And then different story with a different research with our illustrator, because in the first one, when we invited them to join the project, we were kind of like wondering, oh, maybe they want why they want, and then like even, and this is like for free, and then many of them are already in there, like, you know, like one of them are like really famous illustrator. They could easily like make money with like one illustrator, but then they prefer to donate their time to make, you know, like an incredible illustrator for our female stories. So she said, biodiversity is a vital part of my personal interest in climate action. I love reading the stories in this book, and I'm glad that I was able to play a small part in bringing them to life. So this project, in the end, is really highlight a collaboration of women in art and science. So collaboration is like among everything, because even this, we're like 2 different fields, but if we join forces together, then we can make it happen. And so what is our hope? So we, with this book, we basically, yes, of course, we wanted to inspire you by the author's stories, you know, and because those people, they're all still alive until now. Yes, because they wrote their stories, actually. And then so we're not only survive pursuing our dream, you know, to have a career in the herpetology, but we also actually thrive in our field. And we are still here, we're standing firm against all the odds of the system designed to work against people like us. And we really aim, you know, like we really want to create a space where being ourselves is not only accepted, but appreciated, because appreciation is more than acceptance. And we want the world where repression and oppression are fought against and not perpetuated. And specifically, the world where we are all welcome and where we all feel like we belong. So with that, basically the book is for those who blaze their own trails and create their own stories. And feel free to share about the project and buy the book to support the scholarship. And with that, please save the dates, because we will have the virtual book launch around the world. We make it, you know, like really like 5 times, because we wanted to have share this all over the world. So we choose specific times, so then like people all over the world, they can choose like whichever suit their time. And with this, you can like meet Ingrid and then talk about anything to all authors and illustrators, and also the organizer. So basically, like, please feel free to share this, buy the book, and follow our social media as well. And then with it, I would like to say again, thank you, Angelica, to invite me here. It's been a pleasure, and also a pleasure to meet you again. And it's also like, thank you for Raquel and India. It's good to finally like see you, especially in person, because I haven't met you. So yeah, thanks. I hope you're enjoying my talk, and feel free to reach out